I have my phone just as a timer because I have a tendency to. We need to wrap it up. Please get dramatic if you need to. All right. Thank you, everyone, for joining for this session. I really appreciate all of you being here. I know the conference kind of starts early for some of us, so it's great to have everyone here. I am from Western University, so I'm an assistant professor here in the Department of Microbiology and Immunology. And today I'm going to talk to you about a third-year lab course that myself and my colleague, Dr. Ryan Height, who unfortunately couldn't be here today, so I'm sort of presenting on behalf of both of us. But we wanted to share some of the work that we've been doing in our third-year microbiology lab, some of the work that we're doing to sort of move towards more authentic learning and experiential learning in our course. So I wanted to start with land acknowledgement because we are here at Western and I am from Western. I wanted to acknowledge that we are located on the traditional lands of the Anishinaabe, Haudenosaunee, Lenape-Wauk, and Chinookan nations on lands connected with the Township of London and the Sombre Treaty of 1796 and the Ditchwick One Spoon Wampum Covenant. I'd love to chat more about decolonizing microbiology curriculum, but that's not the topic for today. But if anybody wants to chat about that, I'm open to sharing ideas and listening and hearing your thoughts on that. It's something that we're actively trying to do in our department. And I know this isn't necessarily the forum, but I invite anyone who wants to talk about that. Please find me because we're evaluating our curriculum and trying to find ways where we can integrate more perspectives and ways of knowing. All right, so I think for any institution, you have to give some context for what your students do, what their programs look like, because it's different everywhere. It's different at different institutions. It's different in different countries. And so I want to talk a little bit about the BMSc, which is the Bachelor of Medical Sciences program here at Western. So I'm from the Schluck School of Medicine and Dentistry, but we also have a Faculty of Science. So we are separate, although a lot of our students overlap in many of their courses. So a medical sciences student, a student who's accepted into medical sciences as their home program, actually spends the first 2 years of the program mostly taking courses with the Faculty of Sciences, and they don't really get to us until year 3. So we call our students MedSci 1. They're taking foundational courses, biology, chemistry, physics, some electives here and there, but they're really being taught by a completely different faculty than the faculty I am from. And this is where they're learning all that foundational knowledge so that they are prepared to engage in our program in year 3. Second year, again, more foundational knowledge, we get more specified, genetics, cell biology, biochemistry, and it's after MedSci 2 that our students actually have to make a decision about what they want to specialize in. So it's a big decision for our students. They are choosing what path they want to go down. We have a variety of different programs and we call them modules here at Western. So one of the modules would be, microbiology and immunology. We have different ones. We have ones that are joint with other departments, but they're really going through 2 years without really having made that decision, learning just sort of general knowledge from Faculty of Science, a few courses that may come from our faculty, and then they have to make this big decision before they go into year 3. Once they go into year 3, they've made that decision. So we do have a second-year micro-im course that our students can take so that they can get an awareness of what we're all about. And so if they've taken that as an elective, then they would be, maybe keen to join our program. And so in year 3, we have our students sort of specializing. They're going into areas that they're really interested in, the ones that they're passionate about. And then in year 4, there are opportunities for many of our students to do a 4th-year research project in a researcher's lab. Now, not all of our students are able to do that just because we do not actually have enough researchers for every student in our program to do that. So our students will either be considered double majors, where they're sort of specializing in 2 areas, or in an honors specialization program where they will be working in the lab and having to complete a thesis. So those research opportunities for our undergrads are there. However, we are a limited enrollment competitive program, which means the decision of who gets those projects and who doesn't is strictly based on their grades. So it's not ideal, it's not a perfect system. It is the system we're currently working with. And we are working towards having capstone, we're calling them capstone projects, for all students, regardless of module towards the future. But for now, just with the limitations we have with the number of researchers, it's really a 4th-year project that is exclusive to a student who is able to get into an honors specialization module. So this is sort of how our program works. It is sometimes sad for us that we don't get our students until year 3 because we'd love to start having them from the very, very beginning. And it does pose some limitations on how we teach our courses. So for example, when we look at a 3rd-year microbiology lab, we know they've taken biology and chemistry and physics and cell biology, and they've had some lab experience, but they have that lab experience from a different faculty, from other courses that is also preparing students for other avenues that are not necessarily within our discipline. So when we get our students in third year in our laboratory courses, we really have to start building from the ground up. So even though they're in year 3, and they do have a lot of foundational knowledge and foundational skills, we really aren't able to sort of just, assume that they can pipette and assume that they know how to use a vortex or a centrifuge. We do have to start sort of at the beginning to make sure that everybody has the same base knowledge. So that's sort of to give you some context for where our microbiology lab sits. We consider that these students, this is their first micro lab that they're taking, and we are training them sort of from that beginning point. So I want to talk a little bit about our learning objectives, and I sort of consolidated them into 3, and that's just to just sort of minimize how much there is here for you today, but we'd like our students to proficiently apply a range of standard microbiological methods. So we want them learning those foundational skills so that they could go into the workforce, including aseptic techniques, microbial culture, microscopy, molecular biology, and biochemical assays. So even though we are a sort of medical sciences program, we are also trying to prepare our students for careers in industry, because we know that all of them will stay in healthcare or in medicine. We want them to demonstrate strong scientific communication skills through well-structured reports, effective presentations, proper use of scientific language, and active participation in peer discussion. And finally, apply statistical tools to analyze experimental data, assess data reliability and validity, formulate and test hypotheses, interpret findings within the context of existing knowledge, and communicate results clearly using appropriate visual aids. So all of the things you would expect probably from a foundational micro course, but we wanted to change how our micro course was being taught. So previously, it was being taught in an expository format. So I want to chat a little bit about the different types of lab formats you may have seen. Now, we've already heard from our tiny Arthurs a little bit about expository formats with the example of poly. So that's where you're basically having your students run through a protocol. It's a recipe, cookbook, lab, and I think there is time and place for expository labs. So I think especially when it's foundational and you're trying to learn new techniques, very foundational techniques, I think there is still time and place for expository. So who's taken an expository style lab? Lots of people. What did you like about it? Yes.
It was kind of nice to actually see some techniques that we use that we see described before in other classes, but then you actually get to do them for the first time. So
sure. So there's maybe an opportunity to teach more techniques and have exposure to techniques you heard about in lecture, and practice those. Anyone else? Yes.
I think it feels some independence because it makes you think about what you're actually doing, which is a very important process. Really going step by step and making sure everything is logical.
Right. So maybe the cognitive load on a student is quite low because they are just following that recipe. So some good things there. Any other thoughts?
I think for a facilitator who's able to bridge concepts in the lecture to what's happening in the lab, this is really helpful to actually see a process instead of reading it.
Yes, thank you. Yes. One more idea? No?
The success rate is high.
Yes, success rate is high. So there, I know like I can stand here and say like we don't like expository lab formats, but I do think time and place, it is still valuable to our students to sometimes have a moment where they're not overwhelmed with having to come up with all the ideas. So our lab was previously more expository format. It's kind of, those microbial applications books you've seen, you flip through, and like it's literally designed for you. Our course was similar to that. And I think it had practical use and was great for our students to a point. But as we hear about cures, which are also known as allures, I think in Australia, they're authentic, large scale undergrad research experiences, same idea. We are trying to move our course towards that, but also acknowledging that our students are still at a lower level with respect to their laboratory skills. So we were looking at different things we could do. We could go the inquiry route where, we sort of leave it more open-ended. We give them a research question, but then they have to do all the design. We have about 100 students in this course. So then we're looking at resources and the challenges associated with that. We thought discovery, they can come up with their own research question and come up with the experiments they want to do. We can guide it more towards what we're hoping they will achieve. And then of course we have problem-based learning, which is less focused on the experiment you're going to conduct and more on sort of getting to a different end goal. So we were looking at all these different formats in the literature and which one would suit us best for us to sort of adapt our lab style. And we decided on doing sort of what we're calling it a hybrid between expository, discovery, and inquiry. It lands somewhere in that murkiness where we wanted to keep some expository elements in the course so that there are opportunities for students to sit back, hear about something in lecture, and go and perform it in the lab, or just have opportunities to practice their microbial skills without the pressure of getting results or what's the end answer that I have to do or what are you expecting me to do. It's just, we want you to practice. Aseptic technique takes time. We're giving you different experiments to do so you can practice and become proficient at that. And so my example for how we redesigned our course is we went from lots of different Lego blocks that weren't arranged in any sort of format. So each week was a different lab with a different goal and a different technique to trying to form some structure with an overarching project that they are also doing throughout the course. So there are some labs that are more modular or expository, but then we also overlay an entire term project with that they're doing and is integrated into those labs as well. So I really must acknowledge Dr. Brian Haidt. He's the coordinator of the course. He's fearless in education. He will try anything. I'm more reserved. And what about the logistics and how much is this going to cost? And he's dived right in, which I admire him for because he's so brave to do that in education. It's so difficult, especially when you have large class sizes. So he's amazing, and I wish he could be here because he's a great speaker as well and would have so many great things to share. And then also we have Lydia Defoe. Lydia is our technician. We would not be able to do what we do without our technician. And I hope that she wants to work forever because I need her. I need her to stay and continue doing the technical work because she's so fabulous. And she takes all of our crazy ideas and can we do this? And can we have pre-labs and post-lab sessions? And she just runs with it and she's open to that. So I think you need a great team in order to put something like this together. So what are we actually doing? So we have the expository labs. I'm not going to talk about those. They are textbook. You can find them in microbial applications. They're the same ones that you've probably seen in your own labs. But we have decided to go with this biotechnology pipeline. And so essentially we have, for our students, you work for a fictitious biotechnology company called Unitech. Has anyone seen Office Space? Yeah, okay, so Unitech is Bora from Office Space. Our students don't get it, but we chuckle about it. This company specializes in the production of recombinant amylase enzymes, which are used to convert starches and dextrins, so plant-derived sugar polymers, into fermentable sugars for the biofuels and food industries. So we've kind of put an industry flair on this course, sort of to give our students another perspective because they live and breathe medical sciences. And their job is to identify, clone, and assess amylase enzymes for further commercial development. And so we actually want them to go out and find an organism where they can isolate an amylase gene from, clone it, purify it, and then test the enzymatic function. And so we call it their lab pet, not lab pet as in a puppy, but their little bacteria that they're going to go out and sample. So the first thing that they do is they go out, we give them a swab. We do have some intro labs where we do teach them some of the basic microbial stuff. And again, because we don't want to throw them in the deep end, we keep some of those expository labs so they're still learning other things that they'll use later. So we do have a lab where we go out and get water samples from different sources, do a K infusion, and they look at different organisms under the microscope. So we do have some intro laboratories to sort of ease into this, but very, very early on, they are given a swab, a sterile swab, and they're asked to go out and swab something in the environment. We caution them against bathrooms and anything like that. Don't swab your pet. We want them to swab things that may have amylase enzymes. So we encourage them to swab soil, leaves, plants, fungus, something out in the environment is what we're asking them to go out and swab. So they do that, they come in, they plate it. They then need to isolate one of the organisms from their plate. We do try to limit any fungus on the dish by putting cyclohexamide in it because we are asking them to specifically look for bacteria in this instance. So they have to plate out and then they have to isolate and culture it. And throughout the entire term, they have to continue to keep that culture going because we're going to be using it to do different characterization and identify the species. So it's sort of one of their mini projects that they're doing throughout is to keep their lab pet alive and take care of it and subculture it as needed for the assays that we're going to do. Very early, the first thing we do is identify what they've found. So this is with biosafety in mind. We do want them to know what they found. Well, we want to know as soon as possible because for undergraduate labs at Western, we are supposed to be level 1. So if someone were to find a level 2 organism, we would pull it. And we have a bank of organisms now from the couple of years that we've been running this, and we would give them a different organism if needed. But for the most part, I don't think that's really ever happened. For the most part, they pretty much all find level 1 organisms, and it's totally fine. So yes, they identify, so they do 16S ribosomal RNA gene sequencing. We have them use databases, try to identify the genus, the species, all of that to get as close as possible to a match. And then we want them to characterize it. So in addition to doing those sort of generic labs that you see for biochemical characterization of different bacteria, we have them use positive and negative controls that we give them in the lab as if we were going to run an expository lab, but then we also have them test their lab pet. So they're carrying through that lab pet through almost every single lab in the course. I think there's only one lab now where we don't really have anything to do with the lab pet, and it's because we just don't have anything until we get their 16S data back. So they're doing different things, Hervey-Bauer, nitrate reduction, a whole bunch of different tests over a couple weeks. And then once they've done their characterization, we ask them to develop a strategy to clone an amylase gene from their lab pet and into a protein expression vector. So they first have to find the amylase gene, and there's a number of different ones. They have to make sure that it's the catalytic portion of that amylase gene. They need to figure out their cloning strategy. How are you going to design primers? We use the Q5 system, so we have to teach them about that. We talk about if you wanted to just do it with restriction enzymes, how would you approach this? And they have to come up with this strategy, design it, design the primers, we order the primers, and then they do actually clone the gene out, and we put it into a protein expression vector. After that, we then have to have them purify their protein. So previously, we've been histagging the protein with mixed results because histags are not the best in terms of some off targets. So this year we're switching to GST to see if it works any better. But we do have them purify their lab pet protein. Once they've purified it, they have to figure out how much they have, so Bradford assay. And then we set them up to do an iodine starch test. So they do an iodine starch test to see the catalytic activity of the enzyme that they've isolated. And then they have to analyze all of their data and communicate their findings through a variety of different mediums. So we'll talk a little bit about our assessments. So for our modular expository type labs, the students do still fill out worksheets. We've brought this down to a level where the workload on the students is quite low. We really just want to make sure that they understood what they did, the data that they've collected, and putting it into a format. It's sort of practicing putting a figure together. We have some prompting questions in those worksheets, and they just submit those in Gradescope. So it's quite simple for them to do. It's integrated into our LMS. And they're not intended to be a high workload for our students, but we do still want them reflecting on what they've done in those sessions because they are still very valuable, in our opinion. For our biotechnology portfolio, so this is they're working for that company, we do set it up as if they're working for a biotech. When we teach this in class, it's taught like a story. We even have an organizational structure for the company and like all the names are in it and everything. So we sort of play along and have the story unfold as we go through the course. But we asked them to do things that really happen in biotech. So Dr. Height has a colleague who's working in biotech, and he's like, what do you do for reports? What do you write up? What are your deliverables to your boss, to your colleagues? And so we got some templates from them so that we could get an idea of what the real world is doing, because I know in academia, sometimes we don't always see what industry is doing. So we got a little insider help for that. And so we decided to break it down into an executive summary, which they complete early on and encompasses a lot of the biochemical characterization. So they have to do a little bit of a write-up early on. We have them do an elevator pitch. This is our favorite assignment. They get 1 slide, 1 minute, and they have to pitch their experiment. So they have to pitch what they found. So was their enzyme good? Could we use it for commercial purposes? Was it not good? Did your bacteria not grow at a good temperature that would even make it commercially available? So they have to sort of decide, what can I find about my bacteria that's good or bad for me to give a pitch as to whether my biotech company should move forward with this organism in some sort of commercial capacity. So it doesn't really hinge on whether their enzyme works or not. Honestly, when they purify, there's probably so many contaminants that it's going to work. So sometimes we're like, you have no protein and yet you're seeing catalytic activity. That's a bad purification in all likelihood. But we do get them to give this elevator pitch. It's so much fun. Our lab space, we're really fortunate. We have TVs all along the wall, so we can have multiple students doing their elevator pitches to their groups at once. So it's a very lively, fun time. We have them write up a memo, so a shorter document that they have to put together, and then a final technical report that kind of compiles everything. So more focused on technical writing. And you may be thinking, oh, where's your scientific writing? We don't have scientific writing in this course because our students take another lab course, an immunology lab, and we put all the scientific writing into that one. And we did that sort of strategically because there are a few opportunities in our program for students to learn technical writing. So we decided that we would leverage the opportunities we have with this course to teach that to our students and then save the scientific writing for our other lab course that we have. All right, so student feedback. So we get feedback through Western. There's that sort of official thing. Our students aren't great about filling it out. So we do not have great response rates. But at the end of the year, we have part of the course, so there are other assessments in the course that aren't really related to the labs. We have a professionalism grade, participation, those kinds of things. And at the end, they do a self-assessment for that grade. So they actually have to answer a lot of questions like, did I show up on time to labs? Sometimes, always, never. And they have to do that self-assessment as part of the course itself. But at the end, there's a reflection piece where we ask them to talk about their experience in the course, as well as, did you face any challenges or barriers? How did you overcome them? What would you do differently? How would you teach a concept you found difficult to a student taking this course next year? So we tap them for advice on how we can do better. But in that, they often talk about the project. And it's always so interesting because they start with like, oh, so hard. And, my species didn't really have any data in the literature. But then I just realized that I discovered something new. And now, I'm contributing to, science and knowledge. And we don't have them published or anything. I wish we could. But they often talk about the overarching project in that feedback to say that it was a completely different experience that they haven't had. And for our students, because of our system of the year 1 and year 2 being mostly outside our program in many ways, outside of our control, we felt like this was a good opportunity to give our students that won't end up in a 4th year thesis project because they decide to do a double major or to pick a different path, that they still have had an undergrad, thank you, they still have an undergrad research experience. It is somewhat discovery guided. We are providing sort of the path that they're going to take, but we don't know the answers they're going to get. And sometimes the answers they get conflict with the literature, and they have to reconcile why that is. So they might say, oh, the literature says that my bacteria is gram-positive, but in my gram stain, it's gram-negative, or gram-variable, or we're not sure. And they have to sort of problem solve, like where's the discrepancy, what's different? Sometimes with the biochemical tests, there's lots of different ways that you can do those testing for the same thing. So what test did you do? What test did the literature use? Could it be variance in the test or in the environment that you did that? So there's a lot of room. I think for some students, there's a bit of a frustration point partway through with the biochemical tests because not everything lines up perfectly with the literature. But I think it's a healthy frustration that they have. And they, we have sessions, entire lab sessions where our TAs, myself, Dr. Height, Lydia, we are all in the lab and kind of having discussions with them about what did you find and what does it look like in the literature and how can we reconcile the data that you have versus what's out there and how do we put this together? And also at the end, when they have to pitch to their biotech company, a lot of students are like, my amylase didn't work. I'm like, great. What else can you tell me about your organism? Tell me what temperature does it grow at? Does it grow at 56 degrees? Ooh, what can we do with that? So having them see sort of like the different things that they've learned and how they can pitch it still to their biotech company, because they often just see the negative result. And we want them to see that broader spectrum that science isn't always black and white, it's not clean, it's not easy. And failure is a big part of it. And that is how we learn. Laura said that, it's learning. So I think that our students get a lot out of the modules that we put together. And so I want to say thank you to all of you for being here. And I would love if you share your ideas. I also have a link here if you're thinking of doing something like this. This is a little handy design thinking worksheet that you can download and use to try and develop or integrate these types of elements into your own courses. And if you are doing something similar or different at your own institutions, I would love to hear about it because I'm so excited about all the things I've heard, the tiny earthers. I'm calling you tiny earthers now because we're tiny earthers. It's just really great. And it's fun to hear about all the different projects people are integrating to make science more engaging and more fun. So thank you so much.
All the students tenure flings, but you were close. Close. You're very close.
So we have a few minutes for questions, and then I'll try to wrap up quickly, although I suspect.
Two quick questions. How often do you actually get expression from the amylase genes, and how good is the expression actually? Like, do you get big potatoes of expression in your DSPH gels, or?
So the concentration at the end, on average for our students once they purify. Now remember, this is going to be a crude purification. We just do one column, and that's just for time, really, and we don't have an FPLC.
Yeah.
So the average concentration is probably around like 0.8 mg per mil, and they're getting like 0.5 ml to 1 ml off the column. They have lots, and they do a Bradford assay, so some of them will get more, and we will have them scale it back so that when they do their catalytic rate, they can get better numbers. But the purification, like we talked to them about, you know, how accurate is it really?
Yeah.
Because we are just doing the one column, and we do know that for some students who basically, you know, we see that the purification didn't work well because we run an SDS page now, they're still getting sometimes catalytic activity. It's like, well, how do you explain that? So it's not a perfect experiment, but that also gives them something else to think about.
Also, if you're looking at affinity tags, maybe try the strep tag.
Yeah.
Yeah, like it's because I haven't worked with GST, but with a strep tag with the one-step purification, you can get pretty good. Like you can get pretty pure extracts. Yeah, because it's way more specific than a his tag. So
yeah, I'll keep that in mind. Yeah, we were thinking GST just because GST can help stabilize protein.
Yeah, also, yeah, that should help your.
So
yeah, that's true.
we can leave it off. We don't again in terms of time. So it is 26 kilodaltons, but I will think about strep tag because that's another good idea. Thank you.
I have a few questions. So just to continue, just a continuation from that. Are you looking into the specific activity KM and VMAX?
No, so we are not biochemistry. Biochemistry does Michaelis Menten.
Since it is an industry,
yeah,
adding value to what is already out there.
We don't want to put that level of detail on because we know that we don't teach Michaelis Menten and kinetics specifically in our courses that our department offers. Our students are required to take courses from biochemistry as part of their modules. So they're being taught that by another department. And since we don't have like the ownership of how and what is being taught for that, we don't ask them to do we keep it very, very simple for that. The biochemistry lab course, which is offered by the Department of Biochemistry, does do an enzymatic assay where they have to calculate the KM and do all of that. So we leave that to the biochemists. Our students already feel like this is a lot, right? Yeah. Like any lab course, it already feels like a lot. I think if we added like kinetics to this, they might be a rough last lab.
And what's the time period? I'm
12 weeks.
12 weeks.
Yeah.
And are you using universal primers, or are you actually sequencing the entire thing and then designing the custom primers for each and every gene?
For the 16S, oh no, we have individual primers. So everybody, yeah, they design their primers, and we're so mean because we make them do it manually because we want them to understand forward and reverse primers. So they have to actually sit, and we walk them through a tutorial of how to do it, and they have to actually make their primers. And because it's Q5, they have to like have the tail, they have to fully understand how it works. And then later in that lab, after they've designed the primers, we give them software to check it.
Yeah.
You're like, oh yeah, so everybody can now put this in the software and see what you get. And most time, like they usually get exactly the same plus or minus a few nucleotides. So it's very, very close. But then they do find out if they've gone wrong with that reverse primer. It's the tricky one. Yes.
So every fall, every microbiology lab I have a theme.
Yeah.
So last year it was, I went to the Great Salt Lake in Utah and collected a bunch of bacteria. And I gave each one bacterium, and it was unknown. Yeah. So they just characterized that.
That's awesome.
And this year, I isolated bacteria from inside leaves and roots of plants. So they characterized that. My question is, I'm also concerned about the level 1 and level 2. So where do you find that information? Is there a resource that you use?
Well, once we know what organism they have, we search all of them to see, like we look for any information we can find to see if it would be level 1 or level 2. And any that red flag for us, even if we're just not sure, we have, like I said, we have a library from previous years. We have some really cool ones that students have found in the past. And so we've sort of designed, we sort of have like a at any point during the project, if something fails for a student, we have sort of an intervention point where they will still report up until the point where they're stopped working in their stuff. So they still talk about theirs up to that point, but then they would transition to a different organism. We don't make them go back and do all the stuff before that. We provide that data to them because it's already been done. So we do have a fail-safe in the event something weird starts to happen for someone's organism. I don't know, if it doesn't subculture 1 week, we do have something, but we keep everything. That's why Lydia's so great. Her walk-in cold room is like an archive of all the things. And she keeps all the steps from the students. So when a student says, I didn't subculture, she's like, yes, you did, because she's got it all the time. So we do have a system in place to have a backup plan.
Thank you.
You're welcome.
When you're doing the elevator pitches, are the students actively involved with each other?
Yes, and they peer review.
Is there a potential where you could be engaging them as if the company had budget X? I'm wondering if that's a way to help them recognize that actually an outright failure that they can defend the reason we should not pursue this one is actually advantageous to the team. Right.
That's a good point. I don't think we've addressed that before. Definitely something to think about. They have to give them some like marketing and just a little bit of a taste of that so they have an appreciation. They have no concept of how much things cost.
I was just trying to, in hearing that sort of failure thing, like how can we incentivize failure as actually the successful part?
Yeah, we often, it is like, why would you want to move forward with something that has like a disadvantage, right? So sometimes that is what they're presenting is like, we need to stop working with this organism, and this is why. But that's another good reason that our student, we could kind of plug that might be a reason. But yeah, and we do have to give them appreciation for how much things cost. I always tell them when they get their set of pipettes, I'm like, you're holding $2,000 in your hands right now. Multiply that by all the students in the class, we've got a car. Like, it's a lot. It's a lot of, and so I think, yeah, appreciation for how much things cost and whether it's fruitful to move forward is important.
Final question: why amylase? It's because it is easy to analyze.
Because it's easy and they all have it.
Because if you go with same pages, they have a lot of demand. So you can basically collaborate with other companies.
And it's possible, and that's a great idea. Like sometimes we like to switch up what we do in the lab so that the reports from the last year are being moved. So we do sometimes switch things up. So that's a good idea. Another option for us to move to when we've exhausted all the amylases. Thank you. All right.